The organizers for giving me the opportunity to talk here today. I really appreciate it. I'm going to tell you about some of the stuff that we've been doing in our lab. Our lab works on a number of different species, but one of them is the yellow monkey flower, Mimulus guttatus. And we work in lovely locations like this at the Bodega Marine Lab in California. So, first of all, something that's fundamental about biology is that there's often trade-offs that occur when you adapt to different environments. And this is something that we really need to figure out because there is no one organism that dominates in all different niches. And so you can't really be a jack of all trades. And so what happens when you adapt and become good at one thing, and what is the cost that occurs, and how does that cost manifest and prevent you from being good everywhere? So some evidence for this comes from the classic reciprocal transplant experiments carried out by Klaus and Keck and Heisey. And anytime I follow Jill Anderson giving a talk, there's no reason to even do this introduction, but I'll do it anyhow. And so they would do reciprocal transplant experiments, and they'd find that if you transplanted something from like a coastal environment to an inland environment and vice versa, things always did better in their home environment. So things that came from the coast performed best in the coastal environment. Things that came from the inland habitat did best in the inland habitat. And this was true across many plant species. But the question is, how does this happen? What are the genetic mechanisms that are responsible for this? And what are the environmental factors out there in nature that are actually driving this pattern? So in order to get at this, I'm using the yellow monkey flower Mimulus guttatus as a model system. And I'm specifically focused on similar things that Klaus and Keck and Heisey did, which is on these coastal plants versus inland plants. And in our case, the coastal plants are perennials. They live year-round, whereas the inland plants are small annual plants that only live through the winter season that's wet and then die off during the hot summer drought. And so what's driving this is this really strong environmental gradient out in California. So if you've ever been out there during the summertime, you know that San Francisco can be downright miserable. It's covered with fog. And then if you go just inland from there, it can be smoking hot. So when I used to live up in Davis during the summer, I'd come down like every weekend to Berkeley, and I'd always have like a sweater sitting next to me on the car seat. So when I got out of the car in Berkeley, I wasn't freezing to death. But it's a very strong environmental gradient, and it sets up adaptation of all different kinds of species. So back when I was in graduate school, I found that there was this chromosomal inversion that was highly associated with adaptation to these different habitats. So one orientation of this inversion on chromosome 8 was found in all the coastal perennial populations, all in blue, and the other orientation of the inversion was found in all these inland annual populations, all in orange. So in order to test whether this chromosomal inversion was actually involved in adaptation, I spent 4.5 years of grad school making near isogenic lines where I took each orientation of the chromosomal inversion, the coastal one, and stuck it into inland genetic background, and the inland orientation and stuck into the coastal genetic background. And then I planted these out into a reciprocal transplant experiment so we could actually test the effect of the inversion on fitness. So if we see here in the inland habitat, which is Boonville there, you have the inland parent does best, the coastal parent are almost all completely dead because they don't survive to flower in time. And then if you pop in the coastal allele of the inland, or the coastal allele of the inversion into the inland genetic background, you lose fitness, but if you pop in the inland allele of the inversion to the coastal genetic background, you gain a lot of fitness at the inland site. So what's going on here at the inland site, like I mentioned before, is flowering time is crucial. So the inland plants come, they flower earlier, they make more flowers. The coastal plants never make it to flowering, but the inversion itself can have a huge effect on what your timing of flowering and your overall fitness in that habitat. In contrast, at the coastal site, having the chromosomal inversion of the inland type is deleterious. So if you have the inland genetic background, everybody died by the end of the season. They're annual plants. And if you have anything with that inland genetic background, you're toast. But if you have the coastal genetic background, most of the coastal parents make it to the end of the season, so they're perennial. But if you pop in the inland orientation of the inversion, you get a 69% reduction in survival to the second season. So we're having this big effect of this inversion on this annual perennial transition. So overall, what we have is this pattern where we have flowering time is really crucial. The inversion is affecting flowering time, escape from drought in the inland habitat, but it's affecting multi-season survival at the coast. But how does it do that? So that's what I'm going to get into now. So in order to figure out what are the selective agents at the coastal field site, my graduate student, Damian Popovic, set up a really nice experiment where he excluded herbivores as well as salt spray by simply surrounding a plot with this agrofabric material. And he asked, okay, is survival greater of the inland plants at the coast if you surround them and protect them from those two environmental factors? And sure enough, it's dramatically different. So if you look over on the right-hand side, that's what a plot looks like when you don't have agrofabric around it, and pretty much every inland plant is dead. But if you simply put agrofabric around it, almost every inland plant survives and flowers. So we've eliminated natural selection pretty much at this coastal site simply by getting rid of herbivores and salt spray. So overall, what we know now is that the main factors are seasonal drought in the inland habitat and herbivory and salt spray, maybe a couple other factors, but those are the big dominant ones over in the coastal habitat. So what about the genetics? In order to figure out the genetic mechanisms underlying adaptation to these different habitats, when I first got to MSU, I had been thinking about this experiment for a while, and I was like, okay, we've got all these different populations of coastal perennial plants. Let's just grind them up, pull them all together, throw them on Illumina, and then we'll get all the inland populations, throw them on Illumina, and then we'll just look for outliers. And that's what we did. And we got 29 million SNPs. There's something like 5,000 SNPs per dollar. So it's a really nice, really cheap method that other people can use. And what was really great is that we started pulling out all these candidate genes that we would have never been able to get down to otherwise. So one of the key things that came out was that within the chromosomal inversion was a big outlier in GA20 oxidase, which is in a key gene that's involved in the biosynthesis of bioactive gibberellin. And then we also saw that there was evidence for a strong selective sweep that had happened at this gene in the coastal populations. Now, I was really, really excited about this because back in graduate school, I had this idea that gibberellic acid might be involved, and I had sprayed plants with gibberellic acid and found that the inland ones did not respond that much to spraying gibberellic acid, but the coastal ones, if you spray them with gibberellic acid, they turn from having like lateral branches to growing upright, and they expand their internode like in a big way. And it's very similar to the phenotypic effects of intergressing in the chromosomal inversion. So we went back and we did experiments with a bunch more populations and found, sure enough, the coastal populations respond with the gibberellic acid treatment a lot more in terms of the upright aerial branch production when sprayed than the inland populations, which have very little response to the gibberellic acid. So this also got me thinking, like, could there be some kind of thing where gibberellic acid metabolism might play a role in the production of defensive compounds, and could the inversion actually play a role in defensive compound production, because we know that the coastal plants are more heavily defended. And so what was really great was that Greg Howe was down the hall from me, and he was doing all this fantastic work on showing that there's this interaction between the dello genes in the gibberellic acid pathway and the jazz genes in jasmonic acid pathway, and they're kind of antagonistic with each other, so they actually form this teeter-totter-like thing. If GA is up, then JA is down, and vice versa. And we also knew from work that Liza Holesky had done was that the coastal populations were much more defended than the inland populations. So we had this looks like this growth, kind of rapid, growth versus highly defended trade-off going on in the situation. So we used these same near-isogenic lines to ask the question, okay, so is there evidence that the inversion's actually having an effect on this trade-off between reproduction and defense? And so we sprayed the near-isogenic lines with gibberellic acid, and interestingly, there was more of an effect of for the near-isogenic lines that contained the coastal allele of the chromosomal inversion versus the intergression allele where it was just the inland allele. So it looks like it has a pattern that is consistent with the inversion having more of an interaction for the coastal inversion type with gibberellic acid, which suggests to us that, yes, there is some kind of interaction going on here. It may be that the inversions involved directly in gibberellic acid formation, or it is involved in some kind of upstream regulator of gibberellic acid production. What was even more surprising to us, though, was that when we went and looked at defensive compounds, was that we saw that the coastal ones, like we expected, had more production of these phenopropylglycoside defensive compounds than the inland ones, but just simply popping in the inversion allele versus not having the coastal one versus the inland inversion allele, we got very different concentrations of those PPGs as well, indicating that the inversion is also affecting defense in a big way. And so the inversion really is having this effect on this growth defense trade-off in the system as the plants adapt to different environmental conditions. So here's our overall hypothesis of how this works, is that the coastal plants have low GA production, so they're slow to reproduction, slower flowering time, but high JA production and are well defended. In contrast, inland plants have high GA production and have rapid reproduction, but have low GA and defense. So overall, to conclude, there's big differences in environmental variables between habitats. So oftentimes when people are looking at adaptation between habitats, they're often looking at the same environmental variable, but it's really important to look at different environmental variables if you really want to understand local adaptation. Chromosomal inversions play a big role in this system, and since we discovered this inversion, many more systems have shown that chromosomal inversions are important, and that I think future advances in this field are going to be made by integrating our understanding of molecular genetic pathways with these kind of studies in the field. Okay, that's it. Thank you very much.